everyone. Today we will discuss about cell signaling. So as you all know that cell signaling is a process uh, by which one cell will communicate with the other cell within either within the body or with the in the external environment. Okay, so suppose this is one cell and this is another cell, and by using these multiple cells one organ or one body has formed. Okay, so these two cell will communicate with each other to do certain kind of function. So that particular communication is called as the cell signaling. Okay, and in cell signaling there are three stages. Okay, so the first step of the cell signaling is some ligand molecule which is called as the signal molecule. Okay, this is called as the signal molecule. So this signal molecule will firstly so this ligand molecule is also called as the signal molecule. Okay, so this signal molecule will go and bind to the particular kind of receptor, and after binding of ligand to receptor, the second stage is transduction of particular signal to the response side. Okay, so suppose this ligand is coming to activate certain kind of enzyme. So what it will do is ligand will bind with the receptor and transduce this signal until unless this effector molecule will not activated or will not produce any kind of response. Okay, so this is the three stages. First stage binding of the signal molecule to the receptor, and then signal transduction where the chemical signal activated the enzyme. Finally the response is observed. Okay, so these are the three different stages occur in the cell signaling. Then signal molecule can bind to two different kind of receptor. One is the cell surface receptor and another is the intracellular receptor. Okay, so here we can see that in this diagram. Here this diagram, this is the cell, this is the receptor, this green one. Okay, which is present on the cell surface. Okay, and this is the ligand molecule or the signal molecule. Okay, this is the ligand. So this ligand should be hydrophilic in nature because it is present in the outer environment, because it is in contact with the water. Okay, so it should be hydrophilic in nature. Okay, and this type of receptor is called as the cell surface receptor because this is present in the outer surface of the any kind of cell. Okay, and whereas the intracellular receptor is those receptor which is present inside of the cell. Okay, so for this to bind with those particular type of receptor, one suppose this is the ligand. This ligand will carry out by some certain kind of carrier protein. It will carry this ligand molecule from outside to inside of the cell. Okay, after getting inside of the cell, this ligand molecule will bind to the receptor which is present on the nucleus. Okay, and so this type of receptor is called as the nuclear receptor or intracellular receptor because it is present in the inside of the cell. Okay, so this is the intracellular receptor. The ligand molecule in this case should be hydrophobic in nature because it is going inside of the cell. Okay, so it should be hydrophobic in nature. Okay, so some example of ligand which bind to cell surface receptor is epinephrine, non-epinephrine, glucagons, insulin and secretin. Okay, whereas in kind of like in case of intracellular receptor there is cortisol, aldosterone, thyroid, mm, thyroid hormone, retinoic acid, testosterone, and progesterone. Here you can see that most of the ligand is lipid, means it is having the lipid content. Okay, so that's why all the ligand is having lipid content, so that's why it is bind to the intracellular or nuclear receptor. Okay, because it is having lipid characteristic. Okay, whereas these type of ligand are hydrophilic in nature. Okay, then there are different type of cell signaling. First one is the simplest cell signaling is the autocrine signaling. Okay, so what is autocrine? Auto means same. Okay, so autocrine signaling means here the signaling cell and the target cell. Okay, sorry, here the signaling cell and targeting cell both will be same only. Okay, so how, like suppose this autocrine signaling cells that can also bind to the ligand, that itself it is released. Okay, so the signaling cell and the target cell can be same or similar cell. Okay, so here both the cell will be same. Okay, so means the signaling cell and as well as the target cell, both will both the role play by this cell only. Okay, so it will release the signal and it will only receive the signal. Okay, so this kind of signaling is called as the autocrine signaling. Now you are having the paracrine signaling. What is paracrine signaling? Paracrine signaling is a form of cell signaling in which the target cell is near the cell signal releasing cell. Okay, so here means for happening this kind of signaling, two cell be in near to each other. Okay, so the signaling cell will release the signal and the target cell will receive this signal. Okay, so it should be near to each other. Okay, whereas the endocrine signaling, the third signaling, that is the endocrine signaling. In endocrine signaling both the cells will be far from each other. Okay, and the communication will, or the signal molecule will reach to the target cell via the blood stream. Okay, so it is both the cell are far from each other and this signal molecule will go by blood stream to target cell. Okay, so this kind of signaling is called as the endocrine signaling. So these three are the major type of cell signaling, and apart from that there is another kind of signaling which is called as the juxtacrine signaling. So what is juxtacrine signaling? In juxtacrine signaling there should be two cells should be in close contact. Okay, in case of paracrine signaling it should be near to each other, means here it is not necessarily the physical contact or yeah, so in close contact. Okay, but in case of the juxtacrine signaling it should be having the physical contact, or it should be having the close contact with each other via gap junction, via plasmodesmata. Okay, so here in this case it is also called as the contact dependent signaling. Here cells must physically interact to initiate the signaling. Okay, so suppose this is the signal molecule present in one cell. This signal molecule will go from via this channel to the target cell. Okay, and here the receptor will be present and it will sense this ligand molecule. Okay, or bind with the ligand molecule. So here in this case of signal molecule is not free. Okay, but is instead bound to the membrane of the cell. It may interact with a receptor on the membrane of adjacent cell. Okay, so here the ligand is not free, like in case of these three type of signaling. Here the it is bound to the membrane of the cell. Okay, and it will interact to the receptor on the membrane of adjacent cell or the neighboring cell. Okay, so these are the four different kind of signaling. Then like different type of signaling molecule, here also different type of receptor is present. Okay, so the three major receptor is, first one is the ion channel receptor, second one is the G protein linked receptor, third one is the enzyme linked receptor. Okay, so firstly we will see what is G protein coupled receptor. So in G protein coupled receptor the receptor protein, like this is the exterior part and this is the cytosolic part of the G coupled protein receptor, and this is the receptor, okay, which is 7 transmembrane helix, okay. And here once the ligand will bind to this G protein coupled receptor, some conformational changes will occur here, and it goes this signal transduced to the G protein, which is present as a inactive form in the normal condition. When the ligand will bind to this receptor, it will go and activate this G protein, okay. And then this G protein will activate this effector molecule, okay, which can be either adenylate cyclase or phospholipase C or others, okay. So yeah, then after activating effector molecule, it will generate the secondary messenger, that is the cyclic AMP, inositol 3 phosphate, or the DAG molecule, that is the diacyl glycerol, okay. So here this ligand is called as a primary messenger because it is starting the signal cascading, okay. And in the cytoplasm there are 3 different type of secondary messenger is there, which is called as the cyclic AMP and inositol 3 phosphate and DAG, okay. So out of these 3, the GPCR molecule can either release 1 or 2 type of secondary messengers, okay. So this is G protein coupled receptor. Now you are having the ion channel receptor. Ion channel receptor means it is the simplest receptor, okay, where the ligand will go and bind to the receptor, okay. This is the receptor binding side, okay. So ligand binding side in the receptor. So once this ligand will bind to the receptor, the ion channel will open, and the ions will go from outside to inside because of the gradient concentration, okay. So this is the ion channel receptor. Now you are having the tyrosine kinase linked receptor, okay. So what is the tyrosine kinase receptor? This is the tyrosine kinase receptor, okay. So and this is the ligand. Once the ligand will bind to this tyrosine kinase receptor, it will come together and form the dimer, okay. Once it is formed the dimer, it will also autophosphorylate this receptor, okay. It will lead to the autophosphorylation of the receptor, okay. Once the autophosphorylation happen, it activate the tyrosine kinase, okay. This orange color, it's the tyrosine kinase which is present on the cytoplasmic region of the receptor, okay. So here the tyrosine kinase is present, which is present as an inactive form. When the ligand will bind, it will dimerize the receptor and autophosphorylate, lead to the autophosphorylation of receptor as well as the tyrosine kinase, okay. So once the tyrosine kinase will phosphorylate, it gets activated, and it because it is a kind of kinase, so it will phosphorylate the substrate protein, okay. So this is 3 different type of major receptors, okay. Then related to that, you are having a question which is asked in previous year gate, okay. So here you have to match the closest option, okay. So here like receptor tyrosine kinase, cyclic GMP, GTP is activating protein or nuclear receptor, okay. Here you can see the in the group 2 you have to match with the group 2 option, inactivation of G protein, reception of insulin signal, thyroid hormone, or receptor guanyl cyclase, okay. So how you will match? So like in case of the previous slides, we have seen that thyroid is a hydrophobic in nature, okay. So it will go and bind to the nuclear receptor, okay. It will go and bind to the nuclear receptor. So as will be the correct option for as will be 3, okay. So in the given 4 option, only D is having S3, okay. So this will be the correct answer, because thyroid is a signal molecule which is hydrophobic in nature, it will go to the bind to the nuclear receptor. Okay, whereas in the insulin signal, okay, insulin is a kind of hydrophilic molecule, so it will go and bind to the receptor tyrosine kinase. Okay, P2. Okay, because the receptor tyrosine kinase is present on the cell surface. Okay, and so that's why it will bind to the insulin signal. Okay, whereas this GTP is activating protein is the one enzyme which convert GTP to GDP. Okay, so it convert GTP to GDP. Okay, so when GTP is getting converted into GDP, it leads to the inactivation of G protein. Okay, inactive G protein. So it will be vice versa. Okay, if GDP is converting into GTP, okay, so it leads to the activation of G protein. Okay, will lead to the active form of the G protein. Okay, when GTP will convert into the GDP, then it is forming inactive G protein. Okay, when GDP converted into GTP, then it will activate the G protein. Okay, so GTP is activating protein will lead to the inactivation of G protein. Then the remaining thing is the cyclic GMP. Okay, so cyclic GMP is cyclic guanosine monophosphate. Okay, so what is the receptor for it? The receptor for it will be receptor guanyl cyclase. Okay, so this will be the correct form. Okay, so that's all for this class. We will meet in the next class.